Hello everyone. Welcome to today's session. So guys, am I audible? Session. Yes, I am audible. How are you everyone? How are you? And how is your studies going? Because we are continuously meeting, we are providing you daily live classes of all the three subjects. So how is the study going? Is it impacting it? Is it helping you in your studies in any way? Because if yes, then only the purpose is fulfilled of providing all the video lectures of all the important topics here on this particular channel. Okay, so I welcome you all to this wonderful channel called Let's Crack NEET UG in English. I am Aditi Gupta, your biology mentor. And in today's class, I'm going to I'm going to discuss about the semi-autonomous cell organelles. Hi Hiren, welcome to the session. Good evening, bache. So, in today's class, I'll be discussing the semi-autonomous cell organelle. So, as we know that we have started a new topic, and that new topic is not that new now. We have already completed the introduction, the envelope part, and then we have finished the endomembrane system also, right? This unit is really important. Okay, cell, biomolecule, cell cycle, and cell division. 3 chapters are included in this unit, and I'm discussing the first chapter, which is cell. The very basic chapter. We know that life came into existence with the formation of cell, right? So this is the very basic chapter. It is not related to botany nor zoology. It is a basic chapter for the entire biology. And you all must have a sound knowledge, a clear-cut understanding of each and every structure, structural component and function of cell, right? So welcome Ayub, good evening. So come on, without wasting much time, let's start today's topic. But before we proceed, let me introduce myself and let me tell you something really important regarding Unacademy. Okay, so student, my name is Aditi Gupta, and I have been mentoring students for the past 6 years for their NEET UG preparation. Okay, and many students of mine are in their dream medical college. And trust me, guys, now it's your turn. Now it's your turn, and you definitely have to make it right. Now you are well aware of the fact that educators from all across the country, all across the nation are on board, are on this platform, and that too the topmost team of educators who have the experience of training thousands and thousands of doctors, okay, are on this platform. But how can we avail maximum benefit of their knowledge? So you can avail their mentorship, their guidance by getting Unacademy subscription. So let me tell you what is Unacademy subscription and how can you proceed for the same. Okay, students, you need to download Unacademy learning app, or you can visit unacademy.com to buy the subscription. In case if you have downloaded Unacademy learning app, let me tell you something really important. This app hosts some absolutely free of cost features like special classes. See, these free live classes, these are absolutely free of cost. And apart from these live classes, there are also quiz features which are also available absolutely free of cost. So you can attend n number of special classes and n number of quiz sections, right? There is no barrier, and that too free of cost, right? So go learn and explore. Okay, now coming towards the subscription part. So see, after downloading Unacademy learning app, go to the plus or the iconic section, then go to the NEET UG option, then click on get subscription. Well, what are the benefits of having Unacademy Plus subscription? See, you will be getting daily live classes of all the three subjects, physics, chemistry, and biology, along with regular doubt clearing sessions. Okay, along with regular doubt clearing sessions, and the complete NEET UG syllabus will be covered in a detailed manner, in a thorough manner. Okay, along with the well researched PDF notes, and the PDF notes will be provided to you directly by the educator. Then live test and quiz, you will be evaluated, your performance will be evaluated on the basis of live test and quizzes, so that you can check your your progress in your NEET UG preparation journey. Okay, and then you will be able to access all the video lectures, all the PDF notes, and the personalized test papers of all the educators available on the plus platform. If there are 50 plus educators, then yes, you can get access to all the content provided by these educators by the single subscription. And yes, students, you can have one-on-one interaction with your educator. You can ask as many number of doubts as you have. Okay, and Even in case of special classes, even in special classes also, you can have this one-on-one interaction. Okay, I am live daily at 11 a.m. on Unacademy learning app, and the profile link, my profile link is provided in the description box. You can follow me there to get all the updates regarding my class timing. You can have one-on-one interaction with the educator. You can ask your doubts, you can discuss your problems, right? In the Plus, I have discussed all the features. Now let me tell you the important features of Iconic subscription. Unacademy Iconic is a brand new feature which Unacademy has introduced, considering to bridge the gap between the educator and the student. Okay, Unacademy has introduced the feature of personal coach. Yes, dear students, you'll be provided with your own personal coach, which will be there to guide you throughout your NEET UG preparation journey. Okay, which will prevent you from deviating from your path. Then you will be provided with a study planner so that you can utilize your time in a best possible manner. And these study planner will be reviewed to check whether the particular study plan is working on you or not, right? Then you will be provided with in-depth test analysis so that you can check what mistakes you have done in any particular test, go back, rectify them. And in case if the question is being asked in the NEET UG examination, you never repeat the same mistake again, right? It's always good to learn from your mistake, and this is how you learn from your mistake, by doing in-depth test analysis. Okay, then all the benefits of Plus is already also included in the Iconic platform. And in Iconic platform, guys, you will be getting timely motivational sessions from the experts of this field, from the toppers of the NEET UG examination. So you'll be getting timely motivational session also to keep you motivated, to keep you enthusiastic throughout your NEET UG preparation journey. Now, this was about the Plus and the Iconic platform. Let me tell you the fee and how can you proceed for the same. Students, if you have made your mind to go for Unacademy Iconic subscription, where the complete care of a student is ensured, and if you are a 10th pass out student, then go for 2 year of subscription plan. Okay, and if you are a 12th studying or 12th pass out student, then go for 1 year of subscription plan. And the subscription fee is 45,000 for 1 year and 61,000 for 2 year, and that too till 23rd of October. See, the new price list is available. Unacademy price hike was already announced, right? And the new price is applicable from 24th of October 6 p.m. 24th of October 6 p.m. So before this deadline, if you buy your subscription, so for Iconic, see, 1 year, now you have to pay only 45,000 for 1 year, but the fee has been increased to 65,000, applicable from 24th of October. And for 2 year, the fee has been increased to 1 lakh. And we have also introduced a 6-month plan at rupees 38,000. But if you're planning to buy Unacademy Iconic subscription, then go buy it now itself to save your money, right? And get the best available guidance. Okay, and also students, if you use my referral code, which is AG001, you will be getting an instant 10% discount on your course fee. Okay, and this AG001, okay, in this after getting this 10% discount, the applicable fee becomes for 1 year 40,500 and for 2 year 54,900. Okay, similarly for Plus, 1 year fee cost rupees 30,000 and for 2 year the fee is 36,000. But the new applicable fee from 24th of October for Plus for 1 year is 38,500 instead of 30,000. And for 2 year it is 56,000 instead of 36,000. Okay. So if you're planning to buy Unacademy subscription, do not delay it. Okay, because you don't have to pay extra money when you can get the subscription at this much, mere cheaper price. And if you use my referral code AG001, for 1 year the subscription fee will be rupees 27,000 and for 2 year the subscription fee will be rupees 32,400. Okay, students. So come on, this was the detail regarding Unacademy, and this was really an important detail which I provided to you. Now, come on, let's start today's session. So guys, we have discussed about the endomembrane system. We have already discussed about the endomembrane system. Today's class is about the semi-autonomous cell organelle. So what is semi-autonomous cell organelle? We'll be discussing each and every aspect in detail and step by step, right? So autonomous, it means independent. Autonomous means independent. But we are saying that it is semi-autonomous. That means, half independent or partially independent. Okay, that means these are the cell organelles which are partially independent, or you can say partially dependent upon which structure? On which structure? See, I told you that nucleus is the director of cell. That means nucleus control each and every activity of cell. Nucleus control each and every activity of cell. Okay, and you know all the all the cell organelles which are present in the cytoplasm, their activity is being regulated by the nucleus, except for the semi-autonomous cell organelle. What happened in these cases? In these cases, nucleus do regulate their activity, but not in a complete manner. Nucleus regulate their activity in a partial manner. So partially dependent. Okay, in a partial manner. Therefore, I will say that these cell organelles are partially dependent upon the nucleus, and rest of the function they are able to perform by themselves. Okay, then definitely they must be having some structural benefits over the other cell organelles, and that is why they are able to perform their functions on their own, and they are not dependent entirely on the nucleus, right? So now in today's class, I'll be telling you what are those structural modifications which are present in these cell organelles and which are helping these organelles in performing their function on their own up to a certain extent, right? So the first organelle which we are going to study today is the mitochondria, or also known as the powerhouse of the cell. Okay, the powerhouse of the cell, or the ATP mill of the cell. The mitochondria is the most active cell organelle. It is the most active cell organelle and is involved in the process of formation of ATP. Right? So it is one of the semi-autonomous cell organelle. Now, why is it so? So first, let us have a clear-cut look about the structure, or how does the mitochondria structure look like. Okay, see, mitochondria is a double membrane bound structure. Mitochondria is a double membrane bound structure. Let me draw it over here. So mitochondria has two membrane, outer membrane and the inner membrane. Okay, so the membrane of mitochondria is two, outer and the inner. And both the membranes differ in their composition. Both the membrane differ in their composition, right? And their property. So these are what? Outer and inner membrane of mitochondria. And by the presence of two membranes, the structure of mitochondria is divided into two compartments. This, my dear student, is the outer compartment. This is the outer compartment. And this outer compartment is also known as the perimitochondrial space or the intermembrane space, right? Outer compartment is also known as intermembrane space because it is present in between the two membranes, or also known as the perimitochondrial space, right? And the inner compartment, this one is the inner compartment. And this inner compartment is known as the matrix. This inner compartment is termed as what? Mitochondrial matrix, right? So there are two membranes. Then the inner membrane is folded, and it is folded towards the inner side. That means it is infolded. And these finger-like folding structure of the inner membrane is present in order to enhance the surface area. So the presence of this finger-like infolds increases the surface area of the inner membrane. Okay, and these foldings or the inner membrane is termed as cristae. This is termed as what? Cristae. Okay, then what happens? The inner membrane contains some pin-headed particles. You all must have seen a paper pin, right? So it has a needle-like structure and a flat disc-like top. So a pin-headed structure is present on the inner membrane. And these pin-headed particles, structures are known as the F0, F1 particle or the oxysomes. F0, F1 particle or the oxysomes, or the Fernandez-Morin particles. Okay. Then the matrix contain double-stranded circular DNA. The matrix contain what? Double-stranded. DS is double-stranded. Circular, because both these ends of the DNA are tied together. So double-stranded circular DNA is present in the matrix. And also ribosomes are found. Which type of ribosome? So 70S ribosomes are found. So this is a structure of mitochondria containing outer membrane and inner membrane. Now I have shown you the structure, right? Now what happens? See, structural composition of this outer and the inner membrane varies. Okay, it varies. Now, see, mitochondria is a double membrane structure. The structure is shown over here also. The matrix is present, the intermembrane space, then the cristae, the folds, the ribosome, which is 70S type, right? The granules or the stored stored food is present. Then outer membrane, inner membrane, DNA, and the ATP synthase particle, which is the F0 F1 particle. Okay, basically F1 has the ATP synthase activity. I'll be discussing about it in detail. So mitochondria is normally not observed with the help of a microscope until and unless it is stained. So when it is stained with the help of a specific dye, then only mitochondria is visible under the microscope, and the dye is Janus green B. And this is an important question. Janus green B is a specific dye used to stain the mitochondria, right? And the shape of mitochondria, it is also variable. It is also not fixed. It can be sausage shaped to cylindrical or to oval. Newly formed are oval or spherical in structure, right? Then I told you that it is a double membrane bound structure. Now see what happens. The outer and the inner membrane, I'll be writing it over here. In the outer and the inner membrane, there are certain differences, like the ratio of phospholipid to protein. Phospholipid to protein, both are present in the plasma membrane, right? So the ratio of phospholipid to protein in the outer membrane is normally 1:1. That means both are present in approximately equal amount. But the ratio of phospholipid to protein becomes it is 1:3, or we can say that protein is present in more amount. The protein is present in more amount. So here the ratio of protein is to phospholipid becomes what? 3:1. Or phospholipid is to protein becomes 1:3. Anyway, you can read it, right? So protein is present in more amount in the inner membrane of the mitochondria. Then the outer membrane of mitochondria contains a specific type of phospholipid in more amount. And the name of that phospholipid is lecithin. Lecithin. Also known as phosphatidylcholine. Also known as what? Phosphatidylcholine. Okay, so phosphatidylcholine or lecithin is present in more amount in the outer membrane, whereas cardiolipin or also known as diphosphatidylglycerol, also known as what? Diphosphatidylglycerol is present in more amount in the inner membrane, and this is an important point again. I told you, right? So the cardiolipin is present in more amount in the inner membrane of the mitochondria. Now talking about the permeability. So the inner membrane is, selectively permeable. It is, very less permeable. But the outer membrane, it is more permeable as compared to the inner membrane. Because the reason is, in the outer membrane, a specific type of protein channels called porins. Porins are what? These are the intrinsic proteins, and the porins are present in more number as compared to the inner membrane. Okay, so because of the abundance of porins in the outer membrane is sort of more permeable in nature as compared to the inner membrane. Okay, so this was about the structure of mitochondria. Now let me tell you the number of mitochondria. So which factor decides the number of mitochondria in any cell? So first of all, the number of mitochondria is decided or dependent upon the physiological activity of a cell. So that is what I have written over here. Number depends upon the physiological activity. For example, if a cell has to perform active processes, for example, the PCT region of the nephron, it requires high amount of ATP in order to actively reabsorb the substances. So there the amount of ATP will be more, right? So similarly, where more amount of the physiological demand is high, the physiological activity is more, the number will be high. So number can vary. Okay, maximum number of mitochondria is found in the flight muscles of bird. Right? Only one mitochondria is found in certain examples like microasterias, okay, or chlorella fusca. So these are the examples where only single mitochondria is present in their cell, and maximum number of mitochondria is found in the flight muscle of the bird. I hope this point is clear, right? When all the mitochondria of a cell, they are collectively termed as chondriome, All the mitochondria of a cell collectively are termed as what? Chondriome. Okay. And if you remove the outer membrane of the mitochondria, then that structure is known as what? Mitoplast. And that structure is known as what? Mitoplast. When the outer membrane of the mitochondria is removed or absent. Okay. Now this particular slide is for the inner membrane of the mitochondria. Is for the inner membrane of the mitochondria. See what happens. On the inner membrane of the mitochondria, various cytochromes. Cytochromes are what? They are the electron carriers. Cytochromes are electron carriers. So various cytochromes are arranged in a group or in a complex. See, all these structure which is shown over here, complex 1, complex 2, complex 3, complex 2, complex 4. Okay, and in the last is the F0 F1. In the last is the F0 F1. So this particular image is directly from your NCRT. And you will be studying about this image in the respiration chapter, but I have included it here to give you a real feel of how the cytochrome pigments are arranged in the cytochrome carriers are arranged in the inner membrane of the mitochondria. Okay, now see this particular image, the second image. Let me tell you what are these F0 F1 particle or oxysomes, right? See what happens. In the inner membrane of mitochondria, there are present certain pin-headed particles, and these pin-head particles are what? The F0 and the F1. Basically, the F0 F1 particle has two components. The F0 F1 particle has two components. F0 is a proton channel or a proton pump. F0 is what? F0 is a proton channel. See, this F0, it is present embedded in the inner membrane of the mitochondria, and it is permeable to proton, and that too from perimitochondrial space to the matrix region. Okay, that means directional transfer हो रहा है HIN का. Vectorial transfer कहते हैं हम इसे. Vectorial transfer means directional, right? So what happens? F0 allow कर रहा है HIN को from the movement of HIN from the perimitochondrial space to the matrix. Okay, now what happens? Now see what happens. The F0 particle is present in the inner membrane, and then F0 से ही attach है with the help of a stalk-like structure, a round spherical structure which is F1 particle. Now this F1 particle, this F1 has the activity of ATP synthase. So F1 has this activity, ATP synthase की activity है किस पे? F1 पे. So what happens when a proton moves with the help of this F0? है ना, F0 and F1. So what happens? The energy of the proton is utilized in the formation of ATP, and the enzyme present is the ATP synthase. So ये भी F0 F1 particles are present on the inner membrane of the mitochondria. Okay, and these are known as oxysomes or the Fernandez-Morin particle. Fernandez-Morin based on the name of the scientist. So they are arranged on the inner membrane of the mitochondria. Now I gave you an important statement here that cardiolipin is present in the inner membrane of mitochondria in higher amount or in higher quantity. So remember, if it is asked why, although it has never been asked, only the question has been asked that cardiolipin is present in the inner membrane of which of the following cell structure. So we know that it is mitochondria, but I'm telling you the reason why cardiolipin is present in the inner membrane of mitochondria. See what happens. All these complexes, they are arranged together, right? They are known as respirosomes. Together they are known as what? Respirosomes. So for the maintenance of these respirosomes, presence of cardiolipin is required. If the cardiolipin is not present in the sufficient quantity in the inner membrane, then these respirosomes, they are not present in a proper efficient manner. So for the respirosomes maintenance, cardiolipin is needed, and that is why they are present in the inner membrane of mitochondria. I hope this point is clear. Okay, I told you what is mitoplast. When the outer membrane of the mitochondria is removed, then it is termed as mitoplast. And all the mitochondria of a cell together is termed as what? Chondriome. Okay, so this was about the structure of mitochondria. Now, what happens? What are the functions which are related to the mitochondria? See, I told you that mitochondria has two compartments. So talking about the link reaction and the Krebs cycle. What happens in the link reaction? The pyruvic acid or the pyruvate, which has been synthesized by the process of glycolysis in the cytoplasm. Pyruvate has been synthesized in the by the process of glycolysis in the cytoplasm. So these pyruvate are converted to acetyl coenzyme A and CO2. And this link reaction is taking place in the matrix of the mitochondria. Okay, that means enzyme for link reaction is present in mitochondrial matrix. Okay, then what happens in the Krebs cycle? This acetyl coenzyme A, which is being formed in the link reaction, it enters the Krebs cycle and it finally breaks into CO2 and water. So this Krebs cycle is also taking place in the mitochondrial matrix. That means all the enzymes of the mitochondrial matrix are present. All the enzymes of the Krebs cycle are present in the mitochondrial matrix. But is my statement correct? The answer is no. Not all the enzymes of Krebs cycle are present in the matrix. There is an exception. There is an exception. And the exception is enzyme succinic dehydrogenase. Enzyme succinic dehydrogenase. It is the enzyme which is involved in the process of Krebs cycle but is not present in mitochondrial matrix. It is present in the inner mitochondrial membrane. It is present in the inner mitochondrial membrane. Okay, and that is why it is an exception. Rest all the enzyme of the Krebs cycle is found in the mitochondrial matrix, whereas succinic dehydrogenase is found in the inner mitochondrial membrane. Okay, then the electron transport chain. Okay, it runs or it is present in the inner membrane of mitochondria. It is present in the inner membrane of mitochondria. And with the help of these electron transport chain, what happens? Metabolic water is formed and ATP is synthesized. With the help of this electron transport chain, what happens? Metabolic water is formed and ATP is synthesized. Right? All these three steps, link reaction, Krebs cycle and ETS or ETC, takes place in case of aerobic respiration. All these three steps takes place in aerobic respiration. ठीक है बच्चों? Then beta oxidation of fatty acid. जब ये फैटी एसिड ब्रेक डाउन हो रहा है बच्चे, तो बीटा ऑक्सीडेशन ऑफ फैटी एसिड भी किससे हो रहा है? कहाँ पे हो रहा है? माइटोकॉन्ड्रिया के मैट्रिक्स में. इट इज ऑल्सो टेकिंग प्लेस इन द माइटोकॉन्ड्रियल मैट्रिक्स. एंड एज अ रिजल्ट ऑफ बीटा ऑक्सीडेशन ऑफ फैटी एसिड क्या होता है? एसिटाइल कोएंजाइम ए का फॉर्मेशन होता है. एंड दिस इज हाउ द फैटी एसिड एंटर द Krebs cycle, the process of beta oxidation. Okay, so these are the functions which are being performed in the mitochondrial region. And you can see that mitochondria is really important with respect to the aerobic process. Okay, bachcho? Now, moving ahead. Let me tell you something really important. A topic which is related to mitochondrial origin. Okay. We have also not discussed why mitochondria is termed as semi-autonomous. We have also not discussed this point. Why semi-autonomous? So first let me discuss this point. Right? See, be it mitochondria or be it plastid, why are they termed semi-autonomous? Because the reason is, I'm writing it over here. They contain their own DNA. And DNA is generally double-stranded and circular. DNA is double-stranded circular DNA. Both the organelles, mitochondria and the plastid, they contain their own DNA, right? Then they contain their own ribosomes. And ribosome is 70S type, right? So what happens? In a cell, how is the information transferred? So the genetic information which is present in the DNA is transferred to the RNA. And this RNA along with the ribosome helps in the synthesis of protein. So when mitochondria and plastid, they contain their own DNA, they contain their own ribosome, so they will be synthesizing the RNA and then they will be synthesizing the protein. That means, and these proteins will be executing the functions. Okay, that means for the synthesis of protein, the mitochondria or the plastid is not entirely dependent upon the nucleus, and that is why they are known as semi-autonomous. That means half dependent or half independent organelles. I hope this point is clear. Okay, that is why they are known as semi-autonomous cell organelles. Okay, now the question is about their origin. What happens in both the cases, in mitochondria as well as in plastid? Their origin theory suggests that they have been derived, or they have derived from prokaryotic cell. prokaryotic cell. So it is believed that, when the evolution of or the formation of eukaryotic cell was taken place, so a eukaryotic cell, it engulfed a prokaryotic cell. And this prokaryotic cell, it stayed in the cytoplasm of the eukaryotic cell in a symbiotic manner, symbiotic manner. And it was present inside the eukaryotic cell, therefore it is termed as endosymbiotic. And this type of origin theory is known as endosymbiotic origin theory. Okay, and it suggests endosymbiotic origin because the structure of mitochondria or the structure of plastid resemble a typical bacterial cell or a typical prokaryotic cell. See, we have seen that in a typical bacterial cell or a prokaryotic cell, outer cell wall is present, followed by the cell membrane. Rest nothing, no other cell organelle is found except for the ribosomes and double-stranded circular DNA. Only these compulsory structures are present in the bacterial cell. And when you look at the structure of the mitochondria or the plastid, you have seen that mitochondria also has two envelope system. It has two membranes. It also has its DNA, and also, students, the DNA of the mitochondria resembles the DNA of the prokaryotic cell. Not only the mitochondria, the plastid DNA also. The DNA in the organelles, that is the mitochondria and the plastid, has more amount of GC content, and more amount of GC content in DNA is a characteristic feature of prokaryotic DNA. In eukaryotic DNA, more amount of AT content is present. In eukaryotic DNA, what happens? More amount of AT content is present, but in prokaryotic DNA, more amount of GC content is present, and that is what is found in case of mitochondria and plastid also. They do contain ribosome, and ribosome also is of prokaryotic type, 70S, right? So all these structural similarities suggest that they have been derived from a prokaryotic cell. Okay, during the process of evolution, what happened? A eukaryotic cell engulfed a prokaryotic cell, and that prokaryotic cell stayed there for a longer duration of time, and then got modified, and then got converted into mitochondria or plastid. Okay, now what happens during the process of cell division? The new mitochondria are formed by the process of binary fission. See, bacteria also divided by the process of binary fission. Mitochondria are also dividing by the process of binary fission. So this division, it also supports endosymbiotic origin. Got it? Clear? Okay. So this was about the mitochondria. We have seen the structure. We have understood the function. We now know the differences in both the membranes. We now know the importance of each and every component or the compartment. And then we know the origin of mitochondria. Now, we also know what is the reason why is it called as semi-autonomous. Okay. Now, let us study about the plastid. Okay. See, plastid, they are not universal structure. Okay. They are not universal structure. They are also, they are found in the plant cells mainly. And they are, comparatively larger structure. They are easily visible with the help of a microscope. They are easily visible. Like I told, in case of mitochondria were not easily visible, but they are larger in size, and because of its large size, they are easily visible, right? Plastid also is a double membrane bound structure. So see, it is large, double membrane bound structure. Okay, and it is found in all the plants, diatoms, dinoflagellates, and euglenoids. So diatoms, dinoflagellates, and euglenoids. You have studied about these three names in the Protista, kingdom Protista, where all the unicellular eukaryotes are placed, right? So plastid, they contain different types of pigments. And on the basis of presence or absence of these pigments, or in case if the pigment is present, then on the basis of type of pigments, there are different type of plastid which is found in the nature. Okay. What happens? All the type of plastid, they are derived from a common origin, which is the proplastid. So proplastid is the common, ancestral line of from which all the other type of plastid is formed or derived. Okay. Therefore, all the different types of plastids are interconvertible to each other, because the genetic material in all the different type of the plastid is same. Because the genetic material is same, all the different type of plastid is derived from a structure which is termed as proplastid. Therefore, different type of plastids are interconvertible among each other. Okay. Now, see, this is the proplastid. Now, depending upon the requirement of the of the organ, or depending upon the environmental condition also, it can be modified into either chromoplast, okay, chloroplast, or leucoplast. Okay, I'll be covering ethioplast and these three structures later when I'll be having discussions in detail. So the proplastid can be converted into three types of plastid. It can be chromoplast, chromo is color. It can be chromoplast, or the chloroplast, the green color pigment, chlorophyll is present, or the leucoplast. In the case of leucoplast, what happened? The pigment is absent. In case of leucoplast, the pigment is absent. In case of chromoplast, various colorful pigments which are carotenoids are present. In case of chlorophyll or the chloroplast, sorry, it is chloroplast, chlorophyll is present. Okay, so these are the three different types of plastids which are found in a plant cell or in other cells like protists. Okay, now first let us compare the three different types of plastid. Okay, so the chloroplast is green colored. Why? Because it has the pigment chlorophyll in abundance. It has a pigment chlorophyll in abundance. Along with chlorophyll, chloroplast also contain carotenoids. Okay, carotenoids. And then talking about the chromoplast, so the chromoplast are orange, red, or yellow colored pigment. They contain carotenoids, but they do not contain the chlorophyll. Okay, that means the chlorophyll or the green color pigment is present in the chloroplast only. Talking about the leucoplast, so the pigment is absent here, right? And therefore it is colorless. Okay. Talking about the function, so the chloroplast, because of the presence of the chlorophyll, it is helping in the process of photosynthesis. That means with the help of these chlorophyll molecules, what is happening? The sunlight is trapped and it is converted into the chemical energy. In case of chromoplast, what happens? It provides color to the plants. Whereas leucoplast, it serves as the stored food material. It stores food. And based on the type of the food which it has stored, leucoplast is also of different types. Okay. Now, talking about the chromoplast. Okay. It is an important point. Chromoplast stores fat-soluble pigments. It is an important point. It stores what? Fat-soluble pigments like carotene and xanthophylls. Carotene and xanthophylls both are the type of carotenoid pigment. Both are the type of carotenoid pigment. Okay. Yes, bache, that too is present in the case of photosynthesis, and that is why I have mentioned that this chloroplast contain both chlorophyll and carotenoids. The chlorophyll is the main photosynthetic pigment, whereas carotenoid are the accessory photosynthetic pigments. These are the accessory pigments. All the energy which is being trapped by the carotenoids or the accessory pigments are finally transferred to the chlorophyll A molecule or the reaction center. We'll be studying about these in detail in the photosynthesis chapter. Okay, and also Neela ma'am is also discussing the photosynthesis chapter. She might have discussed all these points, right? So the accessory pigments are the carotenoids. Now, see, fat-soluble carotenoid pigments. So carotene and xanthophylls, they are the two types of carotenoid which are found in case of chromoplast. And they are fat-soluble in nature. Okay. Chlorophyll is either absent, or if they are present, then they are present in very less amount. Okay. And it is mainly found in the pericarp. Pericarp is what? Pericarp is fruit covering. Pericarp is what? Wall of fruit or fruit wall. The pericarp has three structures or three layers: epicarp, mesocarp, and endocarp, right? Then petals, okay, and they impart color to them. Because of the presence of the chromoplast, different color is imparted to different structure. But here comes an important statement. Although chromoplasts are present in the petals, but they are present in very less amount. Okay, main color of petal is imparted because of water soluble pigment. So main color of petal is because of the water soluble pigment, and I have already discussed about this particular thing. Water soluble pigment is present in the vacuole. Water soluble pigment is present in the cell vacuole. Okay, in the cell sap, right? Example, anthocyanin, anthochlor, beta-cyanin I discussed. So the main color of petal is because of the water soluble pigment. Okay. So whenever a question comes, you need to bear this point in mind. This is an important point. Do not mark it as chromoplast, because the petal color is responsible vacuole, right? Then what happens? There are certain examples in the nature. See, the carotene, specifically the beta carotene. Okay, Beta carotene is the precursor form of vitamin A. So beta carotene is responsible for the orange color of carrot. Okay, and it is also a precursor of vitamin A. Okay, then the lycopene pigment. It is present. It is responsible for the red color of tomatoes or the chilies. Remember, you know, in the unripened stage, the chili is green. The chili is green. Upon ripening, the chili turns red. Right? Green. That means here chloroplast is present. Upon ripening, it gets converted into chromoplast. It gets converted into chromoplast. So see, this one form is changing into other form. But this reverse is generally not seen. This reverse is generally not seen. Okay. Then lutein and zeaxanthin. These are the examples of certain xanthophylls. These are the examples of certain xanthophylls, and they are present in high amount in the green leafy vegetables. They are present in high amount in green leafy vegetables. Okay, students. So this was about the color body or the chromoplast. Chromo term is for color. Okay, and they provide bright. They are responsible for the bright colors, and the colors are generally red, orange, yellow color. Right now, xanthophylls are mainly yellowish color in range. They are responsible for the yellow color, right? Then, talking about the leucoplast, okay? So, I told you that leucoplasts do not contain plastid, okay? Do not contain pigment, and therefore they are termed as colorless plastid, okay? And since the pigment is absent, okay? They store food or nutrients, and they are generally present in the non-green part of the plant or in the underground part. For example, in the in the root region, they are present in abundance, where they perform the function of storing the food. Okay, so they are present in the non-green part or the underground part, and their main function is to store food. Now, depending upon the type of store food, different name has been, or different type of leucoplast have been identified. Okay, see. The type is amyloplast. Okay. So amylo, you must have heard about amylase, right? So this amylase, like salivary amylase, you have heard? Salivary amylase acts upon the carbohydrates in this, saliva, in the buccal cavity region, right? So this amylo is related to the carbohydrates. Yes, it stores the carbohydrate region. Okay. Then the eleoplast. Eleoplast is related to oil and fat. Okay, so the leucoplast storing oil and fat in their structure is termed as the eleoplast. Alluroplast, it is related to the alluron layer. Alluron layer is a protein rich layer, right? Alluron. So it stores what? It stores protein. Okay, so this is about the leucoplast. What happens in case of leucoplast? All the membranous or structure is absent. It contains a double membrane bound structure, and it contains granules which are storing the food. All the other membranous structures are not found in the leucoplast. Okay, so this was the two type of plastid. Now come, let's see the structure and function of the main or the major type of plastid, which is the chloroplast. Why am I calling it a major type of plastid? Because we know by the formation of chloroplast, what happened? The plant is able to perform the process of photosynthesis, and by this process, plant is behaving as a producer, right? So chloroplast has helped in the process of photosynthesis, right? So what is chloroplast? It contains pigments which are chlorophyll and carotenoid pigments. Okay, chlorophyll are the main pigment molecule, right, which are involved in trapping the sunlight and starting the photosynthetic reaction. There can be different types of chlorophyll A, B, C, D, E, but all the type of chlorophylls, they have a same basic structure. I'll be discussing about the structure also, right? So what is the function? So they trap sunlight required for the process of photosynthesis. Okay, now their number. See, the chloroplasts, they are present in the mesophyll cell of the leaf. Mesophyll cell is the ground tissue of the leaf, right? So the chloroplast is present in the mesophyll cell of the leaf, and normally it is most abundantly found in the mesophyll cell of the leaf, and normally 20 to 40 per cells. That means in a single mesophyll cell, 20 to 40 chloroplast molecules are present. Okay, and the shape of the chloroplast can also vary. It can be lens shaped or oval, spherical, discoid, or ribbon shaped. Ribbon shaped, example in the Spirogyra, right? Discoid, example in the case of Phyllothrics. There are certain examples. Okay, now see, let us know the structure of the chloroplast. So the chloroplast contains two membrane, outer membrane and the inner membrane, and the center region of the chloroplast is known as the stroma. The center region of the chloroplast is known as what? Stroma. Okay, then what happens? single membrane bound structure. Single membrane bound structure called thylakoid is present inside the stroma region. This is what? This is a thylakoid. And these thylakoid are stacked one upon the other like this, like a pile of coin, like this. And this structure, this piled thylakoid structure is the granum. See, it is mentioned over here. So all these in green green structure, this is the granum. Individual, the granum is composed of, the individual disc is the thylakoid. Okay. All these granum are connected with the help of flat membranes. For example, this are connected with the help of flat membranous structure. And these flat membranous structures are known as Fred channels or stroma lamellae. Stroma lamellae. Fred channel, Fred is the name of the scientist. Stroma because it is present in the stromal region, and lamellae is the membrane. So a membranous structure which is present in the stromal part. Okay. Then thylakoid are what? They are flat membranous structures and they are piled up together to form grana. And normally a single grana is made up of 2 to 100 thylakoids. So a single grana can be made up of 2 to even 100 thylakoids, right? Now each chloroplast contains 20 to 100 granum. So each chloroplast, it contains around 20 to 100 these piled structures, and which are connected with the help of Fred channels or the stroma lamellae. Okay. Now what happens? See, the thylakoid membrane, if this is the thylakoid, it contains single membrane. It will be like this. The membrane of the thylakoid will be like this because we know that lipid bilayer is present. Okay, lipid bilayer is present. So like this, the membrane will be covering. So on the inner side of the thylakoid membrane, photosynthetic pigment are present, and that photosynthetic pigment unit is termed as what? Quantazome. Okay, so a group of photosynthetic pigments are present in the inner side of the thylakoid membrane, and that particular point is known as quantazome. Okay, around 230 chlorophyll molecules and around 50 carotenoid molecules. Around 230 chlorophyll and around 50 carotenoid molecules are present. Okay, which forms the quantazome. Okay, this term quantazome was given by Park and Biggins. We'll be discussing about these chlorophyll or the quantazome in detail in the photosynthesis chapter also. Now, the stroma of the chloroplast, it contains enzyme for various processes. First of all, DNA replication enzyme, RNA synthesis enzyme, protein synthesizing enzymes. Okay, so the enzymes are present in the stroma. The photosynthetic enzymes, the enzymes which are involved in the process of dark reaction, are present in the stroma, right? See, rubisco, the most abundant enzyme in the world, or the most abundant protein in the world, rubisco, it is a proteinaceous enzyme, is found in the stroma of the chloroplast, and it participates in the dark reaction of the photosynthesis. It participates in the dark reaction of the photosynthesis, right? So rubisco is present in the stroma of the chloroplast. Then let us have a look. See, I told you this is the thylakoid membrane, and the group of pigments are embedded in the thylakoid membrane like this. And this particular structure which is shown over here is of the chlorophyll molecule. So a chlorophyll molecule has two structure. This particular region is known as porphyrin head. Is known as what? Porphyrin head, or magnesium porphyrin head, or what? Magnesium porphyrin head. Why? Because it contains magnesium ion in the centermost region. Okay, followed by, this is the magnesium ion, see, followed by the 4 pyrrole rings, first, second, third, and the fourth. These are the 4 pyrrole rings, altogether known as the porphyrin ring, right? This is the head region of the chlorophyll, and this is the part which is involved in the absorption of the sunlight. This part is involved in the absorption of the sunlight, while the elongated chain part, this is the tail region. Hydrocarbon tail, right? And this tail region, it only helps in anchoring the porphyrin head to the thylakoid membrane. It is not performing any role in the process of absorbing the sunlight. The main process of absorption is undertaken by the porphyrin head itself. The tail part is helping in anchoring. Okay, anchoring of the porphyrin head. which contains magnesium ion in its centers and contains 4 pyrrole rings, right? So one porphyrin head and second is the phytol tail. Second is the phytol tail. Okay. Then what happens? See, this second chain ring, this second pyrrole ring, and at its third carbon, I'm drawing it here. Okay. So this is the second pyrrole ring. Okay, this is the second pyrrole ring of the chlorophyll molecule. Now at its third carbon, this carbon, what happens at its third carbon? In case of chlorophyll A, CH3 is present. In case of chlorophyll A, CH3 is present at its third carbon. Whereas in case of chlorophyll B, CHO is present. In case of chlorophyll B, what happens? CHO is present at its third carbon. So this is the difference between the structure of chlorophyll A and chlorophyll B. So this is the structure of chlorophyll A and chlorophyll B. I am telling you the basic structure. In detail, we will be studying about the detailed structure of chlorophyll when we will be studying the photosynthesis chapter also, because there will be seeing the relevance, okay, of how is this chlorophyll molecule is formed and their role in light absorption also. But for now, we need to remember that yes, they are present in the thylakoid membrane, and the chlorophyll has two components: porphyrin head, which is actively involved in the process of light absorption, whereas the phytol tail, which is only helping in anchoring, the porphyrin head. And it is mainly made up of what? It is mainly made up of hydrocarbons, and it is lipophilic in nature. The phytol tail is lipophilic in nature. It is well embedded in the inner membrane. See, the plasma membrane is also made of lipid. So lipophilic structure is well accepted by the plasma membrane, and that is why the phytol tail is also lipophilic in nature. Okay. So again, I've told you all the differences between the chlorophyll A molecule and chlorophyll B molecule. This slide is also mentioning the same. This R group, this R is CH3 in case of chlorophyll A, and in chlorophyll B it is CHO. Okay. Then they are soluble in organic solvents. Now, how is this chlorophyll molecule synthesized? So, see, succinyl coenzyme A, an important, intermediate product from Krebs cycle, a process in the respiration, an important intermediate. So, this succinyl coenzyme A along with glycine, it first forms the protochlorophyll, an intermediate form of the chlorophyll. Okay, and then what happens in the presence of light? This protochlorophyll is getting converted into chlorophyll. Okay. Sometimes what happens, if you do not supply light to this protochlorophyll molecule, so a structure which is ethioplast is formed. So in the absence of light, this chlorophyll, it gets converted into a dark color structure which is the ethioplast. If you put back the plant in light, this ethioplast will reconvert into the chlorophyll molecule. Okay. So moving ahead, I have included a certain difference between chlorophyll A and chlorophyll B molecule because it is an important point. You will again be studying these things in the photosynthesis chapter. But since it is important, so I have included it here also. See, the general empirical formula of chlorophyll A is C55S72O5N4 Mg. But in case of chlorophyll, what has happened? An extra oxygen is present in the aldehydic group. So now the formula has been changed. Now it has become C55S7TO6N4 Mg. Okay. When you separate both these chlorophyll molecule with the help of a technique which is paper chromatography. Okay. Chromato is, writing, color, right? And graphy is writing. So you are separating the chlorophyll molecule with the help of color differences. On this paper chromatography, you see that the chlorophyll A molecule is blue-green in color, whereas chlorophyll B molecule is yellow-green, or you can say olive-green in color, right? Chlorophyll A is the primary pigment. It starts the reaction. Therefore, it is also known as reaction center. Whereas chlorophyll B behaves as accessory pigment along with other carotenoids molecule, right? It has a methyl group attached to its third carbon of the second pyrrole ring, whereas chlorophyll B has a CHO group, aldehyde group at the third carbon of the second pyrrole ring. Okay, and then they are soluble in organic solvents, like chlorophyll A is soluble in petroleum ether and it is soluble in methyl alcohol. It is not that relevant to you, but fine, till this point it is important. Okay. Now, I've included a chart. This I've taken from the internet. It is a simple summary chart of, where all the chlorophyll molecules are present. See the chlorophyll A, it is present in the green plants and the cyanobacteria. Chlorophyll B, it is present in the green algae and all the higher plants. Okay. Talking about chlorophyll C, so it is present in dinoflagellates, diatoms and brown algae. Chlorophyll D, it is present in red algae. Chlorophyll E, it is found in xanthophycyan algae or the thallophytes, right? Then in case of bacteria, bacteriochlorophyll A, bacteriochlorophyll B, chlorobium chlorophyll 650 and chlorobium chlorophyll 666 is found. This is 650, this 666 are the absorbents, right? So these are the different type of chlorophyll pigment which are found in the nature. I have summarized all these things in a single table. Okay. Talking about the carotenoids. So the carotene, lycopene, okay, beta carotene or the lycopene. So carotene, it gives orange color. Lycopene, it gives red color. Talking about xanthophyll, it is generally yellow color. But viola xanthan, it is brown. Phycocyanin, okay, phyco is mainly brown, found in brown algae, and it is mainly brown in color. And lutein, I told you lutein, it is found in green leafy vegetables in high amount. Now talking about the phycobilins, these are also pigments. These are also pigments which are involved in the process of photosynthesis, right? So phycocyanin and phycoerythrin are found in case of cyanobacteria. What type of phycocyanin? C phycocyanin and C phycoerythrin. Talking about red algae, R phycocyanin and R phycoerythrin are found in case of red algae. So these are the different types of pigments which we observe in nature. I've tried to include this slide so that in this particular slide you are getting all the summary, right? Now, talking about the origin of plastid. As I told you, plastid do contain their own double-stranded circular DNA, right? They contain their own 70S ribosomes. That means they are able to effectively, synthesize their protein. So they are able to synthesize their proteins. But the enzymes which are participating in the process of photosynthesis, the photosynthetic enzymes are jointly synthesized. The photosynthetic enzymes are jointly synthesized by both nucleus and the chloroplast. Okay. And the plastid is known to have originated from the prokaryotic cell, and that too from cyanobacteria or blue-green algae. You remember in case of cyanobacteria what happened? In case of cyanobacteria, in the cytoplasmic region, membrane-bound structure, thylakoid-like structure were found. And these thylakoid-like structure in the cytoplasm of the BGA or blue-green algae contain the pigment. These pigments were involved in the process of photosynthesis. So the plastid or mainly the plastid is thought to have evolved, or the chloroplast is thought to have evolved from the BGA. Okay, what has happened in the chloroplast structure? This structure has been modified. The structure has been modified, and now the grana has been formed. A more efficient system has been formed by the process of evolution. Okay, so this is how the chloroplast has been evolved. Okay, then what happens? During the time of cell division, the new plastid, they are derived from the pre-existing ones by the binary fission process. Okay, so all these theories suggest the endosymbiotic origin of plastid from the prokaryotic cell. That means during the course of evolution, what happened? A eukaryotic cell, it engulfed a prokaryotic cell or a bacterial cell, and in this case, in case of plastid, a BGA cell, a cyanobacteria, right? And then it stayed in the cytoplasm for a longer duration of time, and then it eventually modified and got converted into other structure, which is known as plastid. Okay, so this was about the structure and function of plastid. Okay, students. Now, let us see what NCERT has to tell us about these two structures. Okay, so mitochondria, also known as mitochondrion, they are thread-like structures, unless specifically stained, are not easily visible under the microscope, and the specific stain is the Janus green B. The name of the specific stain is what? Janus green B. The number of mitochondria per cell is variable depending on the physiological activity of the cell. I told you. In terms of shape and size, also considerable degree of variability is observed. That means there is no constant shape or size of mitochondria. Typically, okay, that means normally it is sausage shaped or cylindrical shape, and the diameter is 0.2 to 1 micrometer on an average. The diameter is 0.5 micrometer and length is 1 to 4 micrometer. This is important. The diameter is 0.2 to 1 micrometer. That means the average diameter of a mitochondria is 0.5 micrometer diameter, right? Talking about the length, so length is in the range of 1 to 4.1 micrometer. Each mitochondria is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, the outer compartment and the inner compartment. The inner compartment is called matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae, and singularly it is known as crista, towards the matrix, that is towards the inside. The cristae specifically increases the surface area, right? The two membranes have their own specific enzymes associated, yes, with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses single-stranded circular DNA molecule, a few RNA molecules, and 70 S ribosomes, and the components required for the synthesis of proteins. And mitochondria divide by fission. So we have discussed about these things. Okay, this is what is written over here. We have already discussed about this thing. I'm just reading the NCERT to make you comfortable that yes, and whatever is written in the NCERT, you know it very well. You have understood about each and every line of the NCERT and even more, right? So this is the diagram which is shown in the NCERT, the outer membrane and the inner membrane, the matrix, and see the inner membrane is folded into finger-like folds which is termed as cristae, and the intermembrane space or the perimitochondrial space, right? Then, talking about the plastids. Plastids are found in all plants and in euglenoids. Okay. See here, dinoflagellates, diatoms are not separately mentioned. But you know that they are also found in dinoflagellates and diatoms. These are easily observed under the microscope as they are large. I told you, right? They bear some specific pigments, thus imparting specific color to the plants. And based on the type of pigments, the plastid can be classified into chloroplast, chromoplast, or leucoplast. The chloroplast contain chlorophyll and carotenoid pigment which are responsible for trapping the light energy essential for the process of photosynthesis. In the chromoplast, see fat term is mentioned over here. This is important. So in the chromoplast, fat-soluble carotenoid pigments like the carotene, xanthophylls and others are present. Okay. This gives the part of the plant a yellow, orange or red color. Okay, because of the presence of these pigments. The leucoplasts are the colorless plastid because they do not contain any pigment, of varied shapes and sizes with stored nutrient. Okay, see the amyloplast stores the carbohydrates. Okay, in plants we know the food is stored in the form of starch, that means they store the starch. Then potato, example potato. Then elioplasts are the oil or the fat storing components, and the aleuroplasts are the protein storing components. Okay, so this is about the plastid. Now let us see the chloroplast. So majority of the chloroplast of the green plants are found in the mesophyll cells of the leaf, and these are of various shapes like lens shape, oval, spherical, discoid, or even ribbon like. And organelles having variable length. Okay, देखिए length is 5 to 10 mm and width is 2 to 4 mm. In mitochondria you saw the length was in micrometer, 1 to 4.1 micrometer, and the average diameter was 0.5 micrometer. But here the length is 5 to 10 mm and width is 2 to 4 mm. Right? So it is comparatively larger in size. After nucleus, the largest component is the chloroplast or the plastid in a cell. Okay. Their number varies. 1 per cell in the chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Okay. And see the structure, the outer membrane, inner membrane. Outer membrane here is more permeable as compared to the inner membrane. Okay, inner membrane is less permeable because the amount of porine is less. Then granum, which is found by the stacking of thylakoid one upon the other, and the stroma lamellae, that means the membranous channel which is connecting the two granas together. Okay. Like mitochondria, the chloroplasts are also double membrane bound. Told you. Of two, the inner chloroplast membrane is relatively less permeable, see, because porine is present in less amount. Right? The space limited by the inner membrane of the chloroplast is termed as stroma. a number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana, and singularly it is known as granum, or the intergranal thylakoids. Okay, that means in between two granas, intergranal. In addition, there are flat membranous tubules called stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoid encloses a space called lumen. Okay, for example, if this is the membrane of the thylakoid, okay, so bilayer membrane is present, so it will be present like this throughout. So this center space is the lumen of the thylakoid. The center space is the lumen. Okay, so this is what it is mentioned here, that the membrane of the thylakoid encloses a space called lumen, and the stroma of the chloroplast contain enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Okay. Then chlorophyll pigments are present in the thylakoids, right? I told you that quantasomes are present on the membrane of the inner side of the membrane of the thylakoid, right? The ribosomes of the chloroplast are smaller, 70S type, resembling the prokaryotes, than the cytoplasmic ribosomes. Okay, and especially the ribosomes which are found in the organelles. The ribosomes which are found in the organelles, that is in the mitochondria or in the plastid, they are also termed as the organellar ribosomes. They are also termed as what? They are also termed as organellar ribosomes. I hope this point is clear. Is it clear, students? Is it clear? So this was about, the semi-autonomous cell organelles. So I told you, since they are not dependent in the, nucleus completely for their requirement of the protein. So therefore, they are known as semi-autonomous. They contain their own ribosome, their own DNA, RNA. They are able to synthesize some of their proteins, and that is why they are semi-autonomous, right? Other important point is the endosymbiotic origin theory, because they are known to have evolved when a eukaryotic cell might have engulfed a prokaryotic cell, because their structure is relevant to a bacterial cell and they do divide by binary fission. So all these structures support their endosymbiotic origin from a prokaryotic cell. So student, this was it. This was for today's class. We have discussed the NCERT also. Okay. In case if you have any doubt, do ask me. Even I have told you I'll be telling you the Telegram link also. I'll be giving you the Telegram link. Okay. Of our channel. You can ask, you can connect with us there also. Let me tell you the Telegram link. I'll be including the slide from next class. The Telegram link, my dear student, is t.me/NEETUG in English. You can connect to us. All the teachers are available there. You can connect to us through this Telegram link. Okay. And in case if you are, if you are confused, see, yes, today at 9 p.m. Let me tell you my timing also. Today at 9 p.m. See, it is not quiz. First of all, it is a practice session. And take maximum advantage of these practice sessions. So at 9 p.m. I'm conducting a practice session. The topic for today's practice session is class 12 chapter number 1. Okay, the reproduction in organism and sexual reproduction in flowering plants. So it will be a quick practice session and the question will be around 30 questions we'll be discussing like yesterday. Okay, now so this is about today's timing. Again, I'll be live tomorrow at 5 p.m. and especially today at 10 p.m. I'm organizing a special session. Today at 10 p.m. I'm organizing a special session, and that too for NEET 2021 preparation strategy at 10 p.m. So back to back I'm arranging the sessions, right? So this one is over at 9 p.m. practice session. Then at 10 p.m. NEET 2021 preparation strategy, just to have a check on you. Right? Again, if you're planning to go buy Unacademy subscription, then do not delay, because from 24th of October from 6 p.m. the subscription price will increase, and you will never want to pay more, right? When even we also don't want you to pay more, right? So go buy your subscription at the cheapest price available. Preference, you can select yourself like for Iconic or the Plus. Okay, and do not forget to use my referral code, which is AG001, to get an instant 10% discount on your course fee. Okay, students. So in case if your timing is clashing, then definitely if there is some time clash, then definitely I'll try rescheduling if any free slot of mine is available. Okay, so thank you.